Hello, everyone. Welcome to the webinar. I'm Ann Thayer, contributing editor to the C&EN Media Group, and I'll be moderating today's event. This webinar is entitled Discovery, Characterization, and Manufacture of Peptide Radiopharmaceuticals and is being sponsored by WuXi AppTec. C&EN works with sponsors to identify topics of interest and value to C&EN's audience and consistent with C&EN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. Just a few technical items to begin. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget or type your query into the Q&A box. If you're disconnected during the webcast, please log in again according to the instructions you received earlier. You're also encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I'll be posing as many as time permits. Please note that C&EN does not endorse any company, products, or services that may be mentioned in the webinar, and that each webinar will be archived at C&EN Online after the webcast. The presentation today is being sponsored by WuXi AppTec. As a global company with operations across Asia, Europe, and North America, WuXi AppTec provides a broad portfolio of R&D and manufacturing services that enable the pharmaceutical and healthcare industry around the world to advance discoveries and deliver groundbreaking treatments to patients. WuXi AppTec's open access platform is enabling more than 6,000 customers from over 30 countries to improve the health of those in need. During the presentation, we'll hear from 4 speakers. First will be Dave Madge, Vice President for Discovery Services at WuXi AppTec. Dave has a PhD in medicinal chemistry from Imperial College London. Our second speaker will be Alex Satz, who has a PhD in organic chemistry from UC Santa Barbara. Alex currently is the Executive Director for DNA-encoded library operations and strategy at WuXi AppTec. Next will be Dr. Jiaji Liao, who obtained his PhD in chemistry at Imperial College London and now leads a peptide synthesis group at WuXi AppTec. And lastly, we'll hear from Xiaowei Wang, Assistant Director of WuXi AppTec's Molecular Imaging Center. Xiaowei did his PhD research in biomedical engineering at the H. Lee Moffitt Cancer Center in Florida. I'll now hand things over to Dave.
Hi everyone, thanks a lot for joining us for this webinar on the topic of radiopharmaceuticals. My name is Dave Madge and I'm part of the Discovery Services team for WuXi AppTec in Europe. Today we're going to be discussing the discovery and optimization of peptide radiopharmaceuticals. I'd like to emphasize that at the end of this webinar we'll have time for a short question and answer session, and of course we'll be very happy to follow up offline with any queries you may have on the topics we discussed today, and you can see my email address at the bottom of this front page. Joining me on this webinar are my colleagues Alex Satz, who will be discussing peptide hit finding, Jiaji Liao will be discussing peptide synthesis and optimization, and Xiaowei Wang will be discussing the evaluation of radiopharmaceuticals, especially with respect to molecular imaging technologies. The field of radiopharmaceuticals has been evolving rapidly, fueled by high-profile, big-value acquisitions, as well as an increasing number of partnerships and investments. In recent years, there's been an increasing focus on small molecules and peptides for targeted delivery of these diagnostic and therapeutic molecules. So, by way of an introduction to the concepts we're going to be discussing today, I show here a cartoon depicting the core elements of a radiopharmaceutical. Working across the graphic from left to right, we have a chelating functional group, typically a large ring containing multiple nitrogen atoms capable of chelating or trapping a radioactive metal atom. The chelator is attached by a linker functional group of a range of different sizes and chemical classes to an affinity ligand for a cell surface receptor. This affinity ligand is tasked with providing selective delivery of the radionuclide to the target cells of interest. The choice of radionuclide deployed may depend on whether the chosen application is to visualize enrichment in the area of high concentrations of the target receptor in support of a diagnosis or targeting therapeutic strategies, or to support the cell-killing delivery of a toxic radionuclide. The huge advantage of this type of molecule is the potential to insert different metal atom radionuclides that can be used for different purposes in different settings. So, for example, we might use the metastable form of technetium-99, a gamma emitter, or copper-64, which is a positron and beta emitter, for the purposes of imaging, cancer cell localization, and tumor size in PET scanning. These radiometals have a relatively long half-life and are well suited to diagnostic applications. However, the same radiopharmaceutical can often accommodate a cell-killing radiometal, such as an alternative isotopic form of the same metal used for diagnosis, or an alternative radiometal such as lutetium-177 or actinium-225. Shall we discuss this aspect in more detail a little later in the webinar? The diversity of chelating capability is illustrated on this slide, with a number of very well characterized cyclic nitrogen containing ring structures able to chelate different radiometals such as those I mentioned earlier. The convenience here is that the insertion of the radiometal is invariably the final step in the synthesis of the radioimaging or radiotherapy molecule. So the cold parent molecule may be prepared on scale and transported to the intended site of use, such as a hospital, for the final step of insertion of short half-life radiometals. Shown here are two examples of such molecules used to treat prostate-specific membrane antigen-positive prostate cancer. Pluvicto from Endocyte, acquired by Novartis in 2018, and PNT2002 from Point Biopharma, recently acquired by Eli Lilly, both of which use the beta emitter lutetium-177 as the radiometal. On this slide, I'd like to briefly illustrate an idealized workflow for the discovery and optimization of a radiopharmaceutical, potentially for diagnostic or therapeutic purposes, and introducing some of the topics we'll spend time on in the next 45 minutes or so. Once we've chosen a target, a highly enriched cell surface protein, we'll identify a ligand for that target, perhaps using one of the many high-throughput screening technologies available to us, but sometimes also starting from a known ligand. And we'll then incorporate a chelating group for the planned radiometal. We use affinity studies to guide us to the most active ligands, but also measure the metabolic stability of the affinity ligand with the linker and chelator attached. Along the way, we'll evaluate different strategies for incorporating the radioligand, and then begin in vivo evaluation for distribution, metabolism, and of course, efficacy. Thereafter, we carry out full toxicological evaluation and develop manufacturing routes before entering clinical development. My colleagues will touch on many of these topics in their presentations. Before I hand over to Alex to discuss peptide hit finding strategies for radiopharmaceuticals, I'd like to briefly comment on the scope of application of peptides as therapeutic agents. And we've been supporting many of these in recent years, not only as standalone therapeutics, but also as targeting and delivery agents for diverse payloads, such as toxins and nucleic acid therapeutics, as well as for the radioisotopes that are the focus of this discussion. So with that, I'd like to introduce Alex Satz to cover the next section of this webinar.
Hello. Yes, my name is Alex Satz, and I'm going to briefly discuss about the different platforms that we have at WuXi to help you find selective ligands against the target of your choice. We have two different styles of platforms that we can do this with. One is DEL, and the other are display platforms. Let me very briefly talk about what the difference is between these two. With DEL, we can generally find ligands that are more synthetic and smaller. We can also find peptides using DEL, where the peptides are also generally smaller in size. And we can also use chemistries that the display platforms can't use. This allows us to incorporate amino acids, which are very distinct and novel, and can form structures which perhaps instead of forming peptides would form more what you might think of as peptide-like structures. And these include macrocyclic rings. For the display platforms, we can use more naturally occurring amino acids, and therefore we can find ligands which correspond to more traditional types of small peptides. This slide shows some of the examples of the different types of DELs that we can offer. On the left-hand side, these are different DELs which we can screen for you. And so you would send your target to us. We need to know what the name of the target is, and we can conduct the DEL screen for you. This includes del-link, and so this is an example of where we can find for you ligands that covalently interact with the target of your choice. This might be needed in cases where the target is considered otherwise an undruggable target, and so its binding pocket isn't necessarily a well-defined pocket. On the right-hand side, we describe different things which we can offer. These are self-service kits. And what that means is that you can conduct the screen within your own lab, and so you don't need to share the name of the target with us. This can be very important for certain companies and certain groups where the target name is very sensitive. I'm going to highlight here the fact that we have a peptide self-service kit. And this means that you can order this kit from us and you can screen it in order to find peptides against your target. Let me briefly talk about what is actually contained within that off-the-shelf peptide DEL kit. It has over 200 billion peptides in it, and they are either of a linear form, such as shown here, or they are the cyclized versions, as shown here and here. As I mentioned before, the amino acids that we can use when making DEL peptides are very different from what can be used for the display platforms. And so we have used over 500 natural, but mainly unnatural amino acids. These macrocyclic rings vary from either 6 to 9 amino acids in total size. The cyclization step itself is through click, and what that means is that the cyclization is a very stable structure. And the total ring size of these are 23 to 32 atoms. We have a classification here of some of the different amino acids that we have used. So as of, for instance, 51 of these have a substitution at the amide NH group. This provides the flow of how these affinities are done and how the workup is also done. And so in the case, for instance, of a self-service kit, where you could conduct the screen within your own lab, you would do the first step, and then you would send the wet lab samples back to us at WuXi, and we would do the other steps up until the point where we would decode the data, and then we would share the results of the screen with you. The risk for you is quite low because you don't need to decide if you want to do, if you want to further work on the results of this screen until you see the decoded data, and you can decide, do you think the screen worked well or not? If you do decide that you feel that the screen worked well, then you can move to the next step and you can unblind the entire data set. A nice thing about DEL data is that the data set's very large, and it allows you to look at not just potentially which peptide ligands are the most potent, but it can also allow you to look at the physical property space of those potential ligands so that you can make the best choice about what hits you would like to follow up, depending upon what the needs are for your project team. And we, of course, at WuXi will help you to look at this data so that you can make the best choices. I would like to highlight a paper that we published about a year back. This was in a collaboration with a company called UNP. And in this case, what we did was we screened the target MDM2 with our macrocyclic DEL pool. And we were able to very quickly discover directly from that selection an extremely potent macrocyclic peptide, which is shown here. And as you can see, it has single-digit nanomolar potency. And in this paper, we also offer or provide a crystal structure of the macrocyclic peptide, which we discovered against the target MDM2. On this slide, we provide a second example of taking the macrocyclic DELs that we have made and running a selection in order to find a potent macrocyclic hit. In this case, we're going after a target which is overexpressed on the surface of fast-growing or cancerous cells, but which is expressed at a much lower level on the surface of otherwise healthy cells. The top panel has a lot of data in it, but what we're showing here is the enrichment level of different potential peptides. Every potential peptide is represented by a single line. And we see that in some conditions where the target is present, the enrichment is very high. And then when the target isn't present, the enrichment drops and is very low. And so what we want to do here is then choose some of these enriched peptides to resynthesize, which is what we did. And we then retest them in their purified form to find out what their binding constants are against the target of our choice. And that's what we have done. And we find that it is indeed a single-digit micromolar potent binder. And I'm going to talk about the results of this and further work that we did with this particular macrocyclic peptide later. But first, I'm going to cover a couple of other different topics in a couple other slides before I return to this. So let me briefly discuss peptide phage display. We also offer this. We offer this in a form where the screening is very similar to what we do with DEL. However, in this case, we're going to be finding potential peptide ligands, which have are more traditional peptides and contain generally naturally occurring amino acids. Usually, when practitioners of this platform run these screens, they narrow them down to the most potent peptides with a focus on potency. We can both do this, but we can also look at a larger quantity of data so that we can focus both on potency, but also take into account the physical property space of these peptides. And then, of course, after we choose what peptides we would like to resynthesize, we can resynthesize them, and we can determine to what extent they are indeed active against the target of choice. This slide highlights all the different ways in which we can determine whether or not the peptides that we find are active against the target, to what extent, and if they are selective. There's a lot on this slide, so I won't go through these one by one, but I will say that we're going to cover in the next couple of slides one example where we use FACS in order to determine the potency of the peptide that we have found. So let me go to that case. So a couple slides prior, we talked about how we had a collection of peptide DELs. We ran a selection against a target which should be overexpressed on cancerous cells and expressed at a much lower level on otherwise healthy cells. We ran this DEL screen, we amplified the barcodes, we read them off, we saw what structures we believe would be potent hits. We resynthesized one of these, and we found that it does indeed have single-digit micromolar KD against that target. We then attached a linker to that peptide with a fluorescent probe, and we can then incubate it with both a cancerous cell line and a healthy cell line. And then we can run those cells through a FACS in order to determine the extent of binding of our peptide to these two different cell lines. And I can show you those results here. We see a great deal of binding to the cancerous cell line, where this receptor is being expressed at a higher level. And in the healthy cell, where the receptor is expressed at the lower level, we see very little interaction of our peptide with the cell surface. And this is determined once again through FACS sorting. We can also look at the uptake of these peptides within the cells themselves. If we look at the data or the panels here on the right-hand side, this is for the healthy cell. We see all these panels remain black, and that shows that there is no particularly strong uptake of this peptide into these otherwise healthy cells. But in the cancerous cells, where this receptor is expressed at a higher level, we see a specific uptake, and we only see that uptake when the peptide is added to the dye. In the controls, we do not see the uptake. And the uptake is shown here by these fluorescent signals, which are now occurring within the cells after the cells otherwise have been washed. Okay, and that's my last slide, and I would like to hand it over to the next speaker.
Thanks, Dave and Alex, for your introduction of RDC drug and the heat screening strategy. I am Jia Zhi. I'm the third speaker here. And in my section, we are going to discuss about the peptide optimization and the synthesis of RDC drugs. Okay, so the first question for us is why peptides? So why we choose peptide as the affinity ligand for RDC drugs? So actually, peptides can make very good affinity ligands, as we can see on the right. So the picture here shows that the peptide can bind to its protein target on a comparatively flat surface. Also, peptides can normally deliver high affinity and high selectivity compared to small molecules. We got example from this publication that one peptide shows single-digit picomolar affinity towards its target, PCSK9 here. So when we compare peptides to antibodies, peptides will normally have a lower cost as well. Okay, so we should ask ourselves the question that how can we obtain such a nice peptide ligand after we get the heat? So probably that's the heat generated by Alex. There are actually multiple approaches or strategies to achieve the peptide optimization. But here today, we will focus on two methods that's widely used. So the first is the single amino acid substitution, and the second is that making cyclic peptides. However, before we can substitute any residues of a peptide, so the problem is that how can we find out where to modify it? So alanine scanning is a way to answer this question. So actually, what alanine scanning basically do is to replace each residue with alanine or d-alanine. So by doing this, we were able to find out the residues that are important for interactions. And it also can tell us the residues that are not important for the interactions. And for the unimportant ones, we can actually change them into some other residues to alter the properties of the peptide, like physical properties or pharmaceutical properties. So here is this example. So the structure is shown here, and this is the sequence. So the cysteine in yellow background is the residues for cyclization. And after alanine scanning, the authors found that the red residues here are important for the interactions, and the C-terminus tail is unimportant. And the black residues here are not making interactions with its target. So with this information in hand, and then researchers can do multiple rounds of a single amino acid substitution. The actual scanning process is much more complicated, but here we will only discuss about two residues as an example. So the first residue we're going to discuss is the phenylalanine here. So when the researchers change this amino acid into the naphthyl substituted amino acid, and they get an increase in potency. And the second residue is the isoleucine here. So when these researchers change the isoleucine to both proline and hydroxyproline, and they both maintain the affinity of the peptide. But they finally choose the hydroxyproline because they believe that the hydroxyl group will enhance the solubility of the peptide. Okay, so after multiple rounds of substitution, and they get their final sequence, so the optimized sequence. So the N-terminus of the peptide was changed to acetyl group. So the acetylation of the N-terminus normally stabilized the peptide. So the C-terminus was deleted. And for the red residues that's making interaction with its target, And they only changed the first one because this is the only first one gave a positive result. There is one thing that's quite interesting. So for the black residues that are not important for any interactions, they were mostly changed to some polar residues. They are D-glutamic acid, homoarginine, glutamic acid, threonine, and hydroxyproline. So by doing this, the solubility of the peptide was greatly improved. And also, the affinity of the peptide was increased from 27 nanomolar to 2.8 nanomolar. So we believe this could be a very good example to show that we can utilize the unimportant residues to increase the solubility of the peptide. So here is another example about making cyclic peptide. So after the researchers get a hit from MRI display, so they did exactly the same thing as the last example. So they did alanine scanning, D-amino acid scanning, and substitution. And then they get a comparatively good peptide. But they are not satisfied with this. So they did something more sophisticated called structural-based drug design. So they get a crystal structure of the peptide binding to its target. So what they found here is that, so these residues are really close to each other in the structure, and they are not making any interaction with the target. So their idea is that probably they can make additional cyclic structures based on these residues. So by doing this, what they hope to do is that they can hold the conformation, hold the structure into a well-defined conformation. So this is what they did. So this is the parent compound, and they make the second cyclization by a RCM cyclization. And then they made the third cyclization by the click chemistry. So we can see that the affinity of the peptide increased significantly from 11 nanomolar to 0.4 nanomolar, and finally to about 8 picomolar with this tricyclic peptide. This is a very good example to show the power of entropy. So with this tricyclic peptide, so there are fewer rotatable bonds in the peptide. That means that when the peptide binds to its target, it will lose less entropy. Okay, so after we understand how peptide optimization can be done, and we have to ensure that we have the corresponding capability. So we know that we need to do alanine scanning, and we need to do amino acid substitution, and we need to make lots of cycles. So in general, what we want is that we can make sufficient analogs for screening. So in order to do that, we can utilize automated synthesizer to achieve parallel synthesis. We can use automated HPLCs to speed up purifications. We have to ensure that we have the optimized conditions as well for difficult coupling and cyclization steps. What's more, so it is better for us to have a large number of unnatural amino acids. So this will guarantee us that we have a large chemical space for the peptide modification. And in some cases, we even have to do custom synthesis for unnatural amino acids to meet our need here. So here I would like to present several case studies to show our capabilities. So the structure on the top left is called Luna 18. So this is a peptide, actually quite hot peptide in phase 1 clinical study. So it's targeting KRAS. We're not going to talking about the function of the peptide, but we are focusing on the structure of the peptides. So as we can see here, the peptide are heavily methylated, and also it contains quite a lot of bulky residues. So these modifications actually gave the peptide additional stability. So similarly, we have an example here to synthesize much more complicated peptides. So these peptides are not only unmethylated, but they are bicyclic. So in order to do that, we have to do coupling optimization. We have to optimize the cyclization site and cyclization conditions. And we also have to optimize the cleavage and purification condition to make sure that our peptides are stable under workup. With all of these optimization, finally we were able to deliver more than 110 heavily modified bicyclic peptides in 3 months with only 6 chemists. Here is another example for RDC synthesis. At the discovery stage, we have a close collaboration of solid phase, solution phase, and purification team. By this collaboration, we were able to obtain more than 1 gram of product in only 1 month with a high purity. And after we optimized the condition, we delivered more than 1 kilogram of the product in 3 months, and also with high purity. What's even better, after the condition optimization, the conversion at each step is high enough so that we can do a single purification of the last step. Okay, so here I would like to show you examples of the general timeline for carrying out either RDC or PDC drugs. As we can see here, our solid phase team can start the synthesis of peptide, and together with the solution phase team synthesizing their payload. Our purification team will be involved to purify any intermediates or crude materials. And after that, we will do the conjugation and the final purification to deliver the peptide. In some cases, the timeline can even be shortened to 3 to 4 weeks. So this shows the benefit of the seamless team collaboration. Okay, so thanks for your listening. I'm now going to hand over to my colleague, Shaowei, who's from our Molecular Imaging Center. Thank you.
Thank you very much for Jazz's presentation. Hello, everyone. I'm Shaowei Wang from Molecular Imaging Center. In this web section, I'm going to talk about efficacy and biodistribution study for peptide radiopharmaceuticals. So at the beginning, I'm going to have a brief introduction about the roles of PET imaging studies in the drug development process. So as we know for drug discovery research, we need to know their target engagement, proof of metabolism, pre-simple actions. So the PET imaging plays an important role in those process. So like the PET imaging result can help with identification of biomarker and targets by comparing patients and the health control, and it can help with monitoring therapeutic responses of a drug candidate. Besides that, it can help with the PK studies by distribution and also specific receptor bindings. More important, PET imaging can help with measuring the metabolisms like the penetration of a candidate through the blood-brain barrier. Besides that, it can help with the clinical development and accounts like the PD action of drug candidates. And also it can help with the biological effects of drug candidates and its interactions with disease conditions. So PET imaging can help with both the PK and PD information in clinic development. As we know, PET molecular imaging is widely applied in the drug development, both preclinical and clinical studies. As the preclinical studies, so the whole flow like the target identifications, validation, radio tracer selection of bias screen, which is help with elect from like 10,000 candidates and get a good result of candidate potential drug candidates, and go through the in vivo testing and target quantifications, and more is PK and biodistribution effective studies. So PET molecular imaging can help with that to provide in vivo imaging and help with the biodistributions, get those data to help with the drug candidate move to the next step, which is the PD and the metabolism actions, and tox study and safety evaluations. The whole process is help with the investigation new drug, which is called IND process. So PET molecular imaging can also apply in clinic trial, which is phase 0 to phase 3. So as we know, for phase 0 to phase 2, we need to figure out the target engagement, the PK study, and more important is ADME. So PET molecular imaging help with it, which is by using the non-invasive in vivo imagings and help to provide ADME basic information, and more is the biodistributions. So it helps with the clinical studies and can help with the candidate drug move to the next step with the phase 3. So after finishes the clinical studies, phase 0 to phase 3, it's called new drug application, which is NDA process approval. So in summary, the PET molecular imaging process plays an important role and widely applied in the new drug development process, both the preclinical and clinical study. So at here in molecular imaging center, both the small and large animal can be done at here. And besides that, the safety evaluation and bioanalysis can be done at here. So I'm going to have a brief introduction about molecular imaging centers. Here is a list about isotope used. We have 20 isotope used here, like the fluorine 18, carbon 64, and zirconium 89, are mostly used for PET-CT imaging. Besides that, we have the therapeutic isotope like lutetium 177, actinium 225, lead 212, and bismuth 213. So those kind of services can be done like in the preclinical research, imaging services, bioanalysis, and toxicity evaluation. So basically, here the new drug development research services can provide two kind of drug. One is the non-radiopharmaceutical, and one is the radiopharmaceuticals. So the idea about the non-radiopharmaceutical is using isotope labeled the tested molecular and perform the PET-CT imaging and also the in vitro analysis. So it helps with provided the PK and biodistribution, even ADME information about the tested compound. Those kind of molecules can be labeled like a peptide and body protein and nanoparticle. So for Radiopharmaceutical, we provide the whole investigation process can be done here, like a labeling method, biodistribution evaluation, PK/PD study, and dosimetries, and for both and the small and large animal preclinical research, like the PET-CT imaging, the whole process it helps with the radiopharmaceutical R&D package. So first, the radiochemical labeling. What can be done is the protein, peptide, body, nanomaterials. So the workflow is the tested compound labeled in the heart cells and then goes through the quality control. After finish it goes through the animal injections, and then the PET-CT imaging is performed. So at here, both over small and large animal PET-CT are available. Both of those scanners have good performance. Besides that, the Pimode Olinda imaging analysis tools is also available. More is we at here we have the gamma counter, which is used for radioactivity quantitative measurement, which is widely used for in vitro tissue quantitative measurement. So I'm going to be talking about the working flow of the efficacy study and biodistributions. So the general idea about efficacy studies, first, it goes to the radio labeling method development. After that, a quality control is the following. We need to make sure the tested compound labeled by isotope is exactly what we need, and with the good stability and the purity, and also have a good radioactivity with a good radiochemical yield. So after that, it can be injected to the animals. Two types of anemometer is available. One is a wild type, and one is pathology model. So for the efficacy studies, the oncology model is widely used. So after that, efficacy measurement is performed. So two kind of ways. One is in vitro tumor evaluations by counting the tumor volumes to get the tumor growth curve and also get the tumor-related volume. And the second way is to do by the in vivo PET imaging by a specific isotope radiopharmaceuticals to visualize the binding to the oncology lesion and also the uptake about the lesion. So when talking about a biodistribution study, which is similar way as the efficacy studies, so first radio labeling and get a good condition about quality control, and then go through the animal injections. Both small and large animal are available, and the wild type and the pathology models are both available. So for large animal biodistribution study, the PET-CT imaging is widely used for the reason is without sacrificing animals to reduce the cost about the large animals, and analyzed by the PMODs with the PET-CT imaging results. And the second way is in vitro gamma counter calculate by sacrifice the animals and collect the tissue and count by gamma counters, which is much more straightforward to get the radioactivity dose, which is widely used in small animal. So here is the biodistribution examples in non-human primates. You can see here we have MIP imaging for zirconia 18 line labeled in bodies for multiple time points from range to 2 hours to 14 days. So at some point we have a dynamic PET imaging for 2 hours. So that's why the vasculatures can be visualized. So we can see the IV veins, the aorta, jugular vein, and the arteries. So it's provided essential information about antibody labeled by zirconia 18 line and goes through with the blood perfusion. With the 10th by, we have the several time points following days. So we can visualize the liver and the biodistributions for total body in the non-human primate. So after get those results of images, we move to next step with the data analysis by the PMOD. So idea of it is the volume of interested organs and delineated. And after a draw of interest organs and the PMOD will automatically provide a result of the biodistribution data of it. So here is just an example about the biodistribution information results. So we have the several time points and also have the several organs which we are interested. Of course, we can add more organs if we are interested to see the biodistributions for a specific target or binding. So that's all about my sections. So appreciate for your attention. I'm handing over to Dr. Dave March. So Dr. March, it's your turn. Thank you very much.
Thanks, Xiaowei, for that overview of the available technologies for visualizing distribution, metabolism, and efficacy of radiopharmaceuticals. I'm going to wrap up with a few words of summary over the next few slides before we move to a question and answer session. We've mentioned several of the approved drugs and drugs under development in previous slides, but I'd like to just use this slide to illustrate some of the diversity of chemistries being used and the relative consistency of the format of the chelating moiety. I'll also mention that alongside the radiometal strategies we've spent most time discussing today, there's an alternative radioligand, fluorine-18, that can be used for imaging purposes and is illustrated here with BMS 986229. This slide provides a high-level overview of the rapid growth of investment, company formation, and deals carried out in this therapeutic space in recent years. In the top left panel, we can see the steep rise in financing in the last few years, fueled by venture capital and IPO, with the bulk of this capital landing in private biopharma companies, as shown in the lower left panel. The upper right panel illustrates the distribution of products under investigation, We focus today on peptide radiopharmaceuticals, but there is also a strong pipeline of small molecule and antibody-directed products under development, and overall around 200 molecules at various stages of discovery or clinical development. This final slide illustrates the diversity of tumor types and targets under investigation, with dozens of different cancer types showing tractability for this class of therapeutic, and around 15 different tumor-specific targets being employed. The annual market for these therapies and diagnostics is projected to reach around $50 billion per year over the next 10 years or so, fueling constant deals at all levels and, of course, several recent high-profile multi-billion-dollar deals. I'd like to finish by thanking you for joining this webinar. Thanks to my colleagues, Alex, Jazze, and Xiaowei, and we're now open for questions, which can be submitted online in the webinar platform.
Great. Thank you, everyone, for a very interesting presentation. We now have time for some questions. Let's start with one for Xiaowei. Xiaowei, which radioisotopes are most favored for radiopharmaceuticals?
I would like to say two kind of one is for diagnosis with for imaging, like fluorine-18, and it's majority used in clinic like FDG and also fluorine-18 related AP45. And also for the therapeutic, we have lutetium-177, which is popular so far. And besides that, the actinium-225 is kind of popular and had a good result for the clinical trial like phase 2. So that's my answer for the isotope used so far widely. And for research, like the preclinical research, the zirconium-18 line is widely used for the molecular labeling.
Okay, great. Thank you for that answer. Going to ask you another one. Someone asked, how important is the peptide thermostability for radiochemical labeling step?
I would say that's a key point for requirement for the peptide, because for the radio labeling, the most important is the heat, because for isotope labeling, we need to break down one of chemical bond and create a new chemical bond. So the heat is a very key point to do that. So that's why we need the peptide have a very good chemostability, so that we can finish that.
Okay, great. Thank you for that. I have another question here. What is the capacity of C14, I-125, or fluorine or H4 radioisotope incorporation?
As we know, for the C14 and H4, they have a longer half-life, like several years or like 10,000 years, which is widely used for the in vitro autoradiography measurements, and I-125, which have the 60 days half-life, which is much longer, which is kind of like similar to the C40 and the H4, unlike fluorine-18, which is very short half-life, about 110 minutes, and is widely used for in vivo non-invasive PET-CT imaging. That's two kind of different types of isotopes. One is for in vitro used, and another one is in vivo non-invasive PET-CT imaging. So that's I think that's the main difference between those radioisotopes.
Great. Thank you for answering that. We have a question for Alex. Alex, can you expand a little on the appropriate choice of hit finding strategies for different target classes?
Sure. So there are a lot of different target classes. So maybe for the point of keeping this answer a bit brief, I'll just talk about two different types. One target class might be a target which resides inside of the cell. And so you would need to find a ligand capable of entering in the cell and then engaging with the target. And for such a hit finding goal, it might make sense then to run a DEL screen in order to find a ligand which is smaller and which has a real chance of being able to get inside of a cell. Now, if your target class resides outside of the cell or is on the cell surface, then you certainly have a wider range of options and a display platform in finding a more natural small peptide or natural-like. That option opens up, and in some cases it might be the better option because usually peptides that are physically larger could be more potent and the selectivity could be higher.
Great. Thank you very much, Alex. We now have a question for Jiaji. How many rounds of peptide design optimization, synthesis, and testing might be required to arrive at a tractable therapeutic?
Well, actually, this actually depends on your target and peptide and how you understand your target. But I listed about 3 or 4 literature in my talk, and for those peptides, it took about 3 to 5 rounds of optimization. And however, this could also be a commercial problem rather than a technical problem. So it depends on how you would like to push your drug candidate to the market. And also, we actually, if we understand the peptide better, we can actually shorten the process for the peptide optimization. And for example, if you've got a hit from a library, normally you won't get a single hit. So you will get several active peptides. So if you carefully analyze the sequence of those positive peptides, probably you will already get some information for the modification you're going to do. So in that way, you can actually shorten the screening method process.
Terrific.
Yeah,
thank you for that answer. I have another question for you and for Alex, I think. Do you think multicyclic peptides will be the trend for future directions, or what do you think about non-symmetric stapling for peptide engineering?
Yes, I definitely believe multicyclic peptides will be a trend, but how can I say that? It has a multiple advantages. So the first thing is that, as I mentioned in my talk, that you've got a fixed conformation. So probably you can treat it as more likely a small antibody rather than a peptide. So it has a quite well defined conformation, so that it has less rotatable bonds. So it has some benefits for the binding. And I don't really understand what does non-symmetrical stapling mean, because almost all type of stapling is unsymmetrical for the peptides that we synthesized.
Alex, do you have anything to add?
I thought that answer was pretty thorough. I would just say perhaps the non-symmetrical stapling means that one ring would be very small and the other ring would be quite larger. And if that is what it means, it's hard to say exactly across all targets if that would necessarily be beneficial. But as has been said, the more rigid these structures are, generally the more potency you will get for the size of the structure. And so with that idea in mind, having these smaller, more rigid structures, I would think, would be something which people would want, and the effect would be one which people would seek after.
Okay. We have another question for Jiaji. Do you have any experiencing synthesizing cis-not peptides?
Can you please clarify what does this not mean? So.
If the person who asked this question is still on the line, maybe they could type something into the Q&A box about what they mean by a cis-not. Don't know that we.
My only understanding of it is that it has multiple cysteine disulfide bridges, and so I think that sort of ties into what the prior question was about. Is the future of these peptides forming multiple bridges and making these peptides more and more stable and rigid? I would say that at least on the side of DEL, that's something that we can certainly do, and we can make dipeptide structures and tripeptides, and we can do a lot of fancy things depending upon what our partners want.
Okay, thank you, Alex, for stepping in there. Just checking here. We have a question about chemical purity. Do you have any insights on how different peptide structures would affect radiochemical purity? Someone mentions they've seen a lot of degradation of peptides. Anyone want to take that?
Hi, Anne, this is Dave. I think in the interest of time, we'll take that question offline. But I see the questioner, and we will provide a comprehensive answer by email. But thank you very much for the question.
Okay. Why don't we wrap up here? If we haven't gotten to anyone's questions, like Dave mentioned, the speakers will get the questions and have them offline and be able to follow up. So thank you again, everyone, for your fascinating presentation. And thank you to all the participants for being a great audience. Please be sure to check CNN or CNN Online for the replay of this webinar and information on the next edition of CNN webinars. Our thanks to On24 for technology and production services. And thank you, WuXi AppTec, for the sponsorship that made this interactive webcast possible. For CNN webinars, I'm Ann Thayer. Hope you all have a good rest of the day.